Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope everyone is fine. I am Asma Kamran. We are continuing the chapter Prokaryotes. Today we will learn about diversity in shapes of bacteria, bacterial spores and cysts, motility in bacteria, structure of bacterial flagellum, genomic organization of bacteria, modes of nutrition in bacteria. Now the first one is diversity in shapes of bacteria. Bacteria exist in three main shapes: spherical, rod-shaped, and spiral. But there are certain bacteria which are capable of changing their shape. They are called pleomorphic. For example, Helicobacter pylori, it can exist in two different forms, helix form and spherical form. So it is a pleomorphic type of bacteria. These are different shapes of bacteria. Spherical are COCCI, rod shape is bacilli, comma shape is Vibrio, spiral shape is spirillum or spirochetes. Now the first one is COCCI. Singular is coccus. They are distinguished on the basis of plane of cell division and the number of cells. If the single cell divide by a vertical division into two cells, it is called diplococci. If it keep on dividing by vertical division into number of cells, so a chain of cells are formed, this is called streptococci. If single cell divide by two vertical divisions, both at right angle to each other, it gives rise to four cells, that is called tetrad. And if it divides at three planes, two vertical divisions at right angle to each other, and the third one is at horizontal plane, then it is called sarcinae. And if it keep on dividing irregularly, it form a grape-like cluster or sheets of cell, it is called staphylococci. These are the examples of cocci. Streptococcus pneumoniae, it is in the form of chains of cells, so that is streptococcus. And Neisseria meningitidis, it is diplococci. Second is bacilli. These are rod-shaped bacteria and they always divide at one plane, that is a vertical plane. If it divide into two, this is called diplobacilli. If it divides into number of cells by a vertical plane or vertical division, it form a chain of cells that is called streptobacilli. There are certain bacilli or rod-shaped bacteria, their ends are spherical, just like cocci, so it is called Coccobacillus. These are the examples of bacilli: Pseudomonas and Escherichia coli or E. coli. Now the third one is spirilla. If it is comma-like, it is called rod-shaped but curved and comma-like, it is called Vibrio. If it is spiral, it is of two types, spirillum and spirochete. Spirillum is when the spiral is thick and rigid, it is called spirillum, and if it is thin and flexible, then it is called spirochete. These are the examples of spirilli. This comma shaped is Vibrio cholerae. This is spirillum minus, and this spirochete is Treponema pallidum. Now these are the different shapes of bacteria, different cocci, bacilli and spiral.
Most prokaryotes are encased in rigid but porous cell walls that protect them from rupturing due to osmotic pressure in watery environments and give different types of bacteria their characteristic shapes. The most common shapes include rod-like bacilli, spherical COCCI, and corkscrew-shaped spirilla. About half of all prokaryotes are capable of directed motion. Spirochetes move in a rolling motion created by a helical wave generated by internal fibrils. Many cyanobacteria and other bacteria glide on surfaces via a mechanism that scientists have yet to fully understand, while some aquatic prokaryotes move up and down in the water by adjusting the amount of gas and vesicles in their cytoplasm. But by far the most common form of locomotion in prokaryotes are flagellum. Prokaryotic flagella may cover the entire cell or form a tuft at one end. Each prokaryotic flagella consists of a single fibril of the protein flagellin, unlike the flagellum of protists, which are complex structures made up of multiple proteins and covered by a cellular membrane. Further, the flagellum of bacteria simply rotate rapidly in their base, as opposed to beating in a whip-like motion as do the flagellum of protists. Recent research has revealed that it is a unique wheel-like structure embedded in the cellular membrane and cell wall that allows the flagellum of prokaryotes to rotate. In fairly uniform environments, flagellated prokaryotes travel randomly. In non-uniform environments, some prokaryotes exhibit taxis, or the capability to move toward or away from a stimulus. In the case of Chemotaxis, prokaryotes move toward chemical stimuli, such as food and oxygen, and away from stimuli, such as chemical toxins. Most mobile photosynthetic prokaryotes exhibit phototaxis, which causes them to move towards light. In some flagellated prokaryotes, magnets formed within the cytoplasm from iron crystals allow them to orient according to the Earth's magnetic field. This allows detection not only of north and south, but also up and down. These magnetotaxic bacteria often live in aquatic sediments and find too much oxygen toxic. Using their unique sensory system, they direct their rotating flagella to carry them into deeper layers of sediment, having the low oxygen and nutrient-rich conditions they require.
Now the spores and cyst. During growth, when essential nutrients are depleted and the conditions like temperature, pH, and the availability of water is not favorable for the bacterial growth, bacteria form resistant and metabolically dormant bodies. There are three types of such structures: endospores, exospores, and cysts. Endospores, as the name shows, they are formed within the cells. They have a complex structure and they're especially resistant to temperature. They are resistant to ultraviolet and gamma radiations, desiccation, that is drying up, enzymes, temperature, and certain chemicals. Exospores are produced outside the cell, and cysts, these are also dormant but different from endospores. These are two types of spores, endospore and exospore. As the name shows, endospores are formed within the cell, that is inside the cell. As you can see over here, they are produced inside the vegetative cell. Exospores, as the name shows, they are formed at the end of the cell, that is on the outer surface of the cell. This is how endospore is formed. Endospores are resistant to high temperature, to the chemicals, to ultraviolet and gamma radiations. These are produced when a vegetative cell, its chromosome divides into two, and the two cells are divided by a septum, so that two chromosomes or two copies of chromosomes get separate from each other. But the division is not equal. Two unequal cells are formed. The larger one is a mother cell. It engulfs the smaller cell or the other cell, and the other cell, it then is retained, or it becomes inside the mother cell, what is removed from that cell, and hard and thick cell wall is formed around it, so it gives rise to endospore. Endospore is resistant to, especially to, high temperature. When the conditions are normal or favorable, it will then start its germination again. Exospores are produced in certain species, and they are produced by budding at one end of the cell, and they are also resistant to desiccation and to heat. But the difference between endospore and exospore is that exospores formation is a part of reproductive process, and they are produced by the process that is called budding, especially in actinomycetes, which is one of the group of bacteria, where the gram-positive bacteria, they form long tubule-like structures called filament, as you can see in this figure. So during their growth, the vegetative cell, when start dividing, it gives rise to spores outside the cell or on the surface of cell. By budding, they form exospores. They form thick cell wall around them and form exospores. These exospores are resistant to desiccation, to temperature, and when the conditions are normal, they start their germination again. The third is cyst. Cysts are produced again when the conditions are not favorable in the species like Azotobacter. They are also dormant cells with thick cell wall. Cyst formation takes place when the cytoplasm of a cell contracts and the cell wall, it becomes thick and hard and rigid around that shrinked cytoplasm, so it gives rise to the cyst. Cyst is also resistant to desiccation and chemicals, but it is different from endospore as it cannot withstand high temperature, means it cannot resistant, it is not resistant to high temperature. So it is different from endospore. Endospores can tolerate high temperature, can survive for many years or for thousands of years due to resistant to high temperature, resistant to pH, etc. But cysts, they cannot tolerate high temperature. Now the next is motility in bacteria. Bacteria, they move with the help of flagellum, and their movement is towards the optimal concentration of nutrients, or maybe away from some toxic substances. So their movement is in response to the chemicals or maybe in response to light. So if they move in response to chemical stimulus, it is called chemotaxis, and if the movement is in response to light, it is called phototaxis. This is chemotaxis. It is a movement of an organism towards or maybe away from a chemical, so that is called chemotaxis. If it is towards a chemical, that is positive, and if it is away from the chemical, it is negative. Now the flagellar movement. Flagellum is a part of bacterial cell, and it is used for locomotion or for the motility of the bacteria. Flagellum is a thread-like structure extending from the surface of the cell. Rods and spirillum such as Pseudomonas, Vibrio, Salmonella, they are motile, but the COCCI, they are usually non-motile because they are without. Now the flagellar movement. This is with the help of flagella, which is a thread-like structure that extends from the surface of the cell, and the distribution or classification is on the basis of number and distribution of flagellum on the cell. When one or more flagella arise only from one or both ends of a rod or spiral-shaped cell, the arrangement is called polar. As you can see, the first one is monopolar monotrichous, as single flagellum arises only at one pole. When single flagellum arises from both the ends or from both the poles, it is called bipolar monotrichous or amphitrichous. If two flagella arise from one pole only, it is called monopolar bitrichous. If two flagella arises from both ends or from both poles, it is called bipolar bitrichous, and also called amphibitrichous. If a tuft of flagellum arises at one end or at one pole, it is called lophotrichous. And if a tuft of flagellum arises from both the ends or from both poles, it is called amphilophotrichous. If the flagella arise randomly over the entire surface of the cell, this is called peritrichous, or the whole surface of the cell is covered by flagellum. This figure is showing many different types of arrangement of flagellum on bacteria. The first one is monotrichous. Single flagella arises from one pole. Second one is amphitrichous. Both poles have single flagella. And lophotrichous, a tuft of flagellum arises from one pole only. Peritrichous, whole of the surface is covered by flagellum. Now the spirochete or spirochetal movement. Spirochetes are the type of spiral-shaped bacteria. They have flagellum, which is in the form of axial filaments, which twist around the spirochete or around the bacterial cell. They are present in a periplasmic space, the space between the cell wall and plasma membrane, due to which they can flex, they can swim, they can creep, and can show many different types of movement. This is a figure that shows the axial filament. You can see these are the filaments which twist around the cell body or bacterial cell and helps the bacterial cell to flex or to creep or to swim. This is a transverse section of the spirochete, and these are the axial filaments which are present in a space that is periplasmic space between the cell wall and cell membrane. This is another figure that shows the axial filament, and this figure, second one, is showing the flagellum which arises from the plasma membrane or cell membrane in a space, periplasmic space between the outer membrane and the cell membrane. Some bacteria like cyanobacteria and myxobacteria such as Oscillatoria, Myxococcus, they show a type of movement that is called gliding movement or gliding motility. These organisms can move slowly over the solid surface. They do not have axial or internal filament like the spirochetes or flagellum on the surface of bacillus cells, but they move on a solid surface by secreting a slimy substance. The exact mechanism is not known, but it is believed that due to the contractile waves or due to the surface tension or maybe by the pushing of the secreted cell line, they glide or they just move on the surface of the substratum or wherever they are moving. As you can see in this figure, this is the slime secreted by the cell, and then they glide on that slime. Now the structure of flagellum, that is bacterial flagellum, different from the eukaryotic flagellum. As you can see that it consists of three parts: ring, hook, and filament. The structure of bacterial flagellum, it is different from eukaryotes because it is without any microtubules and it is non-contractile. It is about 20 nanometer in diameter and up to 20 micrometer in length. It consists of three parts: basal body, which is embedded or present inside the plasma membrane; hook that extends out the surface of the cell; and filament, which is a long structure extends from the hook. The basal body is present in the cytoplasm just beneath the cell membrane. In case of gram-negative, it consists of two rings, and in case of gram-positive, it consists of one ring only. It helps the flagellum to anchor itself in the cell envelope or in the cell membrane. Then from the ring, hook is a curved structure that projects from the surface of the cell, and it connects the basal body and the filament. Filament is a structure that extends or that originates from the hook. It is a hollow structure and is made up of a globular protein called flagellin. Flagellin is arranged in the form of chains that form a helix structure or a helical structure, but it is hollow from inside. So flagellin form chains of helix, which are arranged around the central hollow core. This is a figure that shows the structure of flagellum. As you can see, this is a basal body which consists of rings and that is embedded in the plasma membrane or present just beneath the plasma membrane, originates from the cytoplasm. Then from the ring, hook extends out from the surface, and from the hook, flagellum originates and extends from the surface. This is the difference in the structure of flagellum in gram-positive and in gram-negative. As you can see in the case of gram-negative, there are two rings, but in case of gram-positive, there is only one pair of rings. You can see that in gram-negative, these are two pairs of rings, but in gram-positive, there is only one pair of. Now the mechanism of movement of flagellum. The rotatory movement is produced by the basal body, and it rotates at 360 angle with the help of that pair rings or disc, which enables the basal body to rotate the flagellum. Because flagellum is made up of non-contractile flagellin, so flagellum then rotates and that causes the cell to spin and move forward.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
Now the genomic organization of bacteria. Bacteria have single circular double-stranded DNA which is not enclosed by any nuclear membrane. It is called its nuclear DNA as it controls growth and metabolism. It is also called bacterial DNA as it is present in the bacterial cell. Bacteria, as it have single circular double-stranded DNA, so they are haploid organisms. But during reproduction, there is a brief stage at which they are diploid, means they have 2 chromosomes, or their DNA is duplicated at that stage. They are diploid. Some bacteria also have extranuclear small circular double-stranded DNA, which is called plasmid, and plasmid contain genes for antibiotic resistance. Now the nutrition. Bacteria are also classified on the basis of method of obtaining energy and carbon. They may be heterotrophic, which make their organic compound from other organic sources or compounds, or they may be autotroph, which make their organic compound from inorganic sources like carbon dioxide. As you can see in this figure, they are autotroph and heterotrophic. Autotrophic are again of 2 types, chemoautotrophic and photoautotrophic. Chemoautotrophic are those that synthesize their food with the help of certain chemicals, and photoautotrophic that can use sunlight. Heterotrophic are again of 3 types, parasite or parasitic bacteria, saprophytic and symbiotic. And autotroph are those that can synthesize their food from inorganic substances. They use carbon dioxide as a source of carbon. But the heterotroph, they cannot use inorganic substances, so they obtain their organic food from other organic substances. They cannot use carbon dioxide as a source of carbon. Now the first one is photoautotroph or photoautotrophic bacteria. These are the bacteria which are able to carry out photosynthesis. They obtain their energy from sunlight and use carbon dioxide and water to build up their organic material. These photosynthetic bacteria, they have a chlorophyll which is called bacteriochlorophyll, which is different from the chlorophyll present in eukaryotic plants. Most of these bacteria live in lakes and pools or in the marine environment. They use hydrogen from hydrogen sulfide instead of from water. And their chlorophyll is present in the mesosomes, which are the invaginations of the bacterial cell membrane. These are the examples of photoautotroph, purple sulfur bacteria and green sulfur bacteria. They use carbon dioxide and hydrogen sulfide in the presence of light to build up their organic substance and produce sulfur and water. Now the chemoautotroph. Chemoautotroph are those bacteria that make carbohydrates from inorganic substance, but they do not use light energy. They oxidized inorganic substances such as ammonia, hydrogen sulfide, and methane to make their food. They usually live in soil, produce chemicals such as nitrates, nitrites during the nitrogen cycle, and phosphates, which also help to increase the fertility of the soil. Now the heterotrophic bacteria. Heterotrophic bacteria are also called chemoheterotroph because they cannot make organic compounds from inorganic sources. They depend on small or large molecules which they have to absorb. There are three types of heterobacteria: heterotrophic bacteria, saprotrophic or saprobic, parasitic, and symbiotic. The first one is saprotroph. Saprotrophic are those bacteria that live and get their nutrients by digesting the dead bodies of animals and plants or the waste which is produced by the animals. These are also called decomposing bacteria. Number 2 is parasites. Parasitic bacteria are those that live inside the bodies of other living organisms, and these living organisms are called host organisms. So they obtain their nutrient from their host, and in return they damage or they are harmful to the host organism, and they cause many different diseases in the host, so they are also called pathogen. For example, Streptococcus pneumoniae. That is all about for today's lecture. Your assignment is that number 1, differentiate between archaea and bacteria. Give general characters of cyanobacteria. Differentiate between gram-positive and gram-negative cell wall, and describe different shapes of bacteria. Thank you very much.